Assalamu alaikum. Today we want to talk about chapter 3, amino acids and peptides, and we will cover the following sections. 3.1 amino acids are three-dimensional. 3.2 structures and properties of amino acids. 3.3 amino acids can act as both acids and bases. The peptide bond, and finally small peptides with physiological activity. Now, amino acids are three-dimensional. Among all the possible amino acids, only 20 are usually found in proteins. The general structure of amino acids include an amino group, an H2 functional group, and a carboxyl group, COH functional group, that dissociate to give the carboxylate anion, COO-, and a hydrogen ion. Now, as you can see here, we have this carbon. This is called alpha carbon, and to this carbon is bonded a carboxyl group and an amino group. And then we have R. R is the side chain, and then H is always there. So this carbon is designated alpha carbon. So this amino group is designated alpha amino group, and this is called alpha carboxyl group. Now the R group is the one that is different in all the 20 amino acids. So the R group determines the identity of your amino acids. So we have alpha carbon, as I said, side chain, and then we have the two-dimensional formula shown here can only partially conveys the common structure of amino acids, because one of the most important properties of these compounds is their three-dimensional shape or stereochemistry. Now, we have two stereoisomers of amino acids, the L and D amino acids, based on similarity to glyceraldehyde. And stereoisomers are molecules that differ from each other only in their configuration. Now, structure and properties of amino acids. Now, except for glycine, all amino acids have at least one chiral center, the alpha carbon, and are chiral stereoisomers. The vast majority of alpha amino acids have the L configuration at the alpha carbon. And proline is usually in the L form. Now, the side chains of carbons in amino acids other than glycine are designated with the Greek symbol starting at the alpha carbon. Notice here, as we said, all naturally occurring amino acids have the L configuration. However, you can find the amino acids in bacterial cell wall and in some antibiotics. And D and L is important. For example, a sugar molecule with the D orientation may be sweet, whereas the L isomer is bitter. And the same for a drug. A drug that is helpful in one orientation may be poisonous in the other orientation. So again, amino acids can be frequently referred to by three-letter or by one-letter abbreviation of their names, with the one-letter designation being much more prevalent. And table 3.1 lists these abbreviations. So if we look at this table here, we see all the 20 amino acids here, and with a three-letter abbreviation, and this is the one-letter abbreviation. Notice that the three-letter abbreviation is almost the first three letters of the name. Ala for alanine, arg for arginine. We have some exceptions. For example, asparagine ASN to distinguish it from aspartic acid ASP, and also glue for glutamic acid and glen for glutamine to distinguish them from each other. Gly for glycine, his for histidine. Isoleucine is an exception. Leucine, lysine, methionine, phenylalanine, proline, serine, threonine, tryptophan is also an exception. Tyrosine and valine. What is required from you is the three-letter abbreviation. We will not ask you about the four-letter abbreviation. Now, in order to study amino acids correctly, they are grouped into the following groups. We have nonpolar amino acids. One group of amino acids has nonpolar side chains. This group consists of 8 amino acids. They are named here. 9 amino acids. Okay. Alanine, valine, leucine, and isoleucine contain aliphatic hydrocarbon group. Proline has an aliphatic cyclic structure, and it's referred to as an amino acid. In phenylalanine, the hydrocarbon group is aromatic. In tryptophan, however, the side chain contains an indole ring, which is aromatic. So both phenylalanine and tryptophan are said to be an aromatic amino acids. In methionine, the side chain contains a sulfur atom in addition to aliphatic hydrocarbon groupings. And in this figure shows you the structures of the commonly found of amino acids commonly found. These are the 9 we mentioned. These are the 9 amino acids that we mentioned. And you should know all these during your study. You should memorize all the structures of these non-polar amino acids. Now, then we have a second category, polar neutral amino acids. Another group of amino acid has polar side chains that are electrically neutral at neutral pH. This group includes serine, threonine, tyrosine, cysteine, glutamine, and asparagine. These are also 6 amino acids. So 6 plus 9, 15, and 15 remains. Now, the amino acid glycine is sometimes included here for convenience since it lacks a nonpolar side chain. Glycine was mentioned in nonpolar amino acids, but sometimes it's included in polar neutral amino acids. Why? Because it lacks a nonpolar side chain. Now, in serine and threonine, the polar group is a hydroxyl group bonded to aliphatic hydrocarbon groups. Now, in tyrosine, the hydroxyl group phenol is bonded to an aromatic hydrocarbon group. Notice that here we have tyrosine, also another aromatic amino acid. In cysteine, however, the polar side chain contains ethyl group and SH. Glutamine and asparagine contain amide groups in their side chains. And here you can see the 6 polar amino acids that we mentioned. Note here, tyrosine is similar to phenylalanine, except that it has an OH on the aromatic ring. So we can say that phenylalanine is a precursor of tyrosine. Now 5 amino acids remains from the 20. We said 9 are non-polar, 6 are polar and polar but uncharged, and we have 5 remains. 2 of them are called acidic amino acids. These are glutamic and aspartic acid. They have carboxyl groups in their side chains, in addition to the one present in all amino acids. A carboxyl group can lose a proton, forming the corresponding carboxylate anion. Now, because of the presence of the carboxylate, the side chain of each of these 2 amino acids are negatively charged at neutral pH. And this is the reason why they are known as acidic amino acids, because they have negatively charged R groups at physiological or at a neutral pH. And these are the 2 acidic amino acids. You see, they are similar to each other, except that here the R group of glutamic has an extra CH2 group. Now the other 3 amino acids are classified as basic amino acids. These are histidine, lysine, and arginine. They have basic side chains, and the side chain in all 3 is positively charged at or near neutral pH. That's why they are referred to as basic, because they have positively charged R groups at or near neutral pH. Now in lysine, the side chain amino group is attached to an aliphatic hydrocarbon tail, as we will see. In arginine, the side chain basic group, called the guanidino group, bonded to an aliphatic hydrocarbon tail, is more complex in structure than amino group. Now in free histidine, the pK of the side chain imidazole group is 6, which is not far from the physiologic pH. So that's why they said, no, histidine is the only amino acid with buffering action at physiologic pH. Here you look, this is, if we start with lysine, this is the alpha carbon, this is beta, gamma, delta, epsilon. So there is an amino group here, the second amino group. This is called epsilon amino group to distinguish it from the alpha carbon amino group. Now arginine, this has a guanidinium group, and histidine has the so-called the imidazole group, as you can see here. Now after that, we can look at this example here. Structures and properties of amino acids. In the following group, identify the amino acids with nonpolar side chains and those with basic side chains. Alanine, serine, arginine, lysine, leucine, and phenylalanine. And second question, the pK of the side chain imidazole group of histidine is 6. What is the ratio of the uncharged to charged side chains at pH equal 7? And we can have the solution here. Amino acids with nonpolar and basic side chains. Nonpolar, alanine, leucine, and phenylalanine. Basic are arginine and lysine, as we said. Now serine is not in either category because it has a polar side chain. It is polar but uncharged. Now the ratio of uncharged to charged side chains at pH 7 is, this is the ratio. If we apply the Henderson-Hasselbalch equation, pH equal pKa plus log A minus concentration divided by HA concentration, then the ratio will be 10 to 1. Why? Because the pH is 1 unit higher than the pKa. So in addition to the common amino acids, we have the so-called uncommon amino acids. These are derived from the common amino acids, produced through post-translational modification when the parent amino acid is modified after the protein is synthesized by an organism. Now we have three examples. We have hydroxylysine and hydroxyproline. These are found only in a few connective tissue proteins such as collagen. So this is your proline and this is your hydroxyproline. And you have lysine and this is your hydroxylysine. The third example is that thyroxine, which comes or derived from thyroxine. Then tyrosine is a precursor of thyroxine, which is found only. Thyroxine is found only in the thyroid gland. Now, amino acids can act as both acids and bases. Now, in a free amino acid, the carboxyl group negative and the amino group positive are charged at a neutral pH. Now, amino acids without charged groups on their side chains exist in a neutral solution as isotherms with no net charge. Isotherm has equal positive and negative charges in solution. It is electrically neutral. Now, neutral amino acids do not exist in this form without charged groups. Okay, titrations of amino acids. Let's look at this figure here. This figure shows us the ionization of amino acids. Now, what happens when we titrate an amino acid? When an amino acid is titrated, its titration curve indicates the reaction of each functional group with a hydrogen ion. Now, we usually start with the fully protonated form. If we take, for example, this general form for an amino acid where R is a non-polar and F polar uncharged, we start with fully protonated form. The net charge is +1, so it is called cationic form. Then we start titration. OH- will abstract a proton from the carboxyl group. We titrate this group first because it has lower pKa, so it is a stronger acid. And then we get this form called isoelectric isotherm, where the net charge is 0. Then we add a second proton, or a second mole of OH-, which titrate this alpha amino group. And we get this anionic form with a net charge -1. So as you can see here, we have three species here, or three forms. Now, form 1, if we call this the cationic form, predominates at a pH lower than 2.34, and form 2 predominates at a pH higher than 2.34 and lower than 9.69, and the anionic form predominates at a pH higher than 9.69. Now, if you plot or you construct a titration curve for this, you will get the following titration curve. So when an amino acid is titrated, its titration curve represents the reaction of each functional group with the hydrogen ion. Now then, this is the titration curve of alanine, and it is of a diprotic acid. Now we start with the fully protonated form here. We add OH minus, the pH increases until we reach here at midway titration, where the acid form and the conjugate base form, they exist in equal amounts, and this pH will equal pKa1. Then we continue adding the second half. Now we reach here at the isoelectric pattern here, the second form. Then we started with the fully protonated form, then 50-50, and then we have the isoelectric pattern. Now we fully titrated the alpha carboxyl group. Then we titrate the alpha amino here. We add another mole of OH minus, pH increases, and at midway titration here at 1.5, we have again pH equal pKa2 this time. Here the two forms are in equal concentration, and then we continue adding the second half mole so that the pH increases and we get our anionic form. So we start with the fully protonated form, then isoelectric pattern, then anionic form, as you can see here. Now we call this inflection point, as you remember in the previous chapter. So alanine has 2 pKa values, so it has 2 inflection points. So it has 2 inflection points. Now if we see the next figure here, it is the titration curve of histidine. Now, and it is that of a triprotic acid. Now histidine exists in, it has 4 ionizable forms. Form 1 fully protonated, then we have form 2. We start with a +2 charge, then +1, then 0, then -1. And since it is a triprotic acid, it has 3 pKa values and it has 3 inflection points. Now let's define the so-called Pi. Pi is the pH at which the net charge on the amino acid is 0, or the number of positive charges equal the number of negative charges. It can be determined from the plot, as you can see here, or it can also be calculated using this table. This is the pKa values of common amino acids. In the free amino acids, alpha carboxyl and alpha amino groups are titrable groups. As you can see, these are the values. Do not memorize any of these pKa values because they will be given to you. Now, amino acid isoelectric point. Now, the pH at which a molecule has no net charge is called the isoelectric pH, or isoelectric point, Pi. And Pi, this property can be put to use in separation methods. Now, the Pi of an amino acid can be calculated by this equation. Pi equal pKa1 plus pKa2 divided by 2. Now, Pi for glycine falls midway between the pKa values of the carboxyl and amino groups, like this. You will be given these two values. You add them, divide by 2, and you will get, this is the Pi, or the isoelectric point of for alanine, or for glycine. This is for glycine. You can calculate for any of the 15 amino acids with the same formula, the 9 non-polar amino acids and the 6 polar but uncharged amino acids. Now, example, given the following amino acids, aspartic acid, alanine, arginine, which of these amino acids has a net charge of +2 at low pH, which has a net charge of -2 at high pH? Arginine and lysine have net charges of +2 at low pH because of their basic side chains. Aspartic acid and glutamic acid have net charges of -2 at high pH because of their carboxylic acid side chains. Alanine and leucine do not fall into either category because they do not have titrable side chains. What is the pI for histidine? Now the numbers to substitute in the equation for the pI are 6 pKa1 and 9.1 pKa2, which gives a pI of 7.58. So this is the pI. So if for the 5 amino acids, the 3 acidic, the 3 basic, for the 3 basic, you add pKa1 plus pKa2 divided by 2. For the acidic amino acids, you add pKa1 plus pKa2 divided by 2. Now the peptide bond is a bond which is formed between individual amino acids where one water molecule is eliminated. It's an amide bond between amino acids in a protein. Individual amino acids can be linked by forming covalent bonds. Bond is formed between the alpha carboxyl group of one amino acid and the alpha amino group of the next one. Water is eliminated in the process and the linked amino acid residue remain. As we will see in the next figure, a bond formed in this way is called the peptide bond. So, peptides are molecules formed by linking 2 to several dozen amino acids by amide bond. So as you can see here in this figure, formation of the peptide bond. This is the alpha carboxyl of one amino acid and this is the alpha amino of another amino acid. They approach each other. They react with the elimination of one water molecule to form this is called the peptide bond. Now, polypeptide chain, backbone of a protein formed by linking amino acids by peptide bonds. Figure 3.9. Small peptide showing the direction of the peptide chain. We have, if you see here, we have R1, R2 to R6. We have 6 amino acid residue connected together by 5 peptide bonds, as you can see. Now, this is the direction of the peptide chain. It is from the N terminal to the C terminal residue. So this is, for example, called hexapeptide, contains 6 amino acids with 5 peptide bonds. It has an N terminal end and a C terminal end, and the direction is always from N to C. Now a question about the peptide bond. Is the peptide bond carbon-nitrogen bond? Is a peptide bond is usually written as a single bond with one pair of electrons shared between the two atoms. Now single bond can be written as a double bond with a shift in the position of pair of electrons. Now we have resonance structures, structural formulas that differ from each other only in the position of electrons. Peptide bond can be represented as a resonance hybrid of two structures, and peptide bond has a partial double bond character, making it stronger than a single bond, and rotation is restricted. So we can see here, you see here, this is not a single bond and a pure single bond, and this is not a pure double bond. Because of these moving of electrons, we have these, we call these resonance structure of the peptide bond, and due to this there is no free rotation about the peptide bond. However, we have a free rotation between this carbon and nitrogen, and this carbonyl carbon and this alpha carbon. So from this side and from this side, alpha carbon with nitrogen, alpha carbon with carbonyl carbon, we have a free rotation here. So we can say that the peptide bond is rigid and planar. Why? Due to resonance stabilization. So again, there is no free rotation around the bonds between the alpha carbon of a given amino acid residue and the amino nitrogen and the carbonyl carbon of that residue, but there is no significant rotation about the peptide bond. Now this stereochemical constraint plays an important role in determining how the protein backbone can fold. Now finally, the final section here, we have small peptides with physiological activity. Now, what are some biological functions of small peptides? One of the most important of these roles is that of hormones, and we will see many examples of hormonal control as we discuss biochemical reaction in the later chapters. Of course, other classes of compounds, particularly steroids, can be hormones also. The notoriety of steroid hormones in doping scandals in sports can obscure the fact that peptides also can be hormones. We can take a quick look at peptide hormones now to prepare the way for later discussions. Now here we have some important peptide hormones have cyclic structures. Now two well-known examples with many structural features in common are oxytocin and vasopressin, as you can see here. These are the their structures. Now in each there is an SS bond responsible for the cyclic structure between cysteine residue number 1 and cysteine residue number 6 in both of them. This is one point. Each of these peptides contain 9 amino acid residue. Then you can count from 1 to 9. From 1 to 9. So they should contain how many? 8 peptide bonds. Each has an imide group rather than a free carboxyl group at the C-terminal. So we have, this is N-terminal, but at the C-terminal end we have an amino group, not a carboxyl group, as it should be usually. And each has a disulfide link between cysteine residue position 1 and 6. Now the difference between these two peptides is that oxytocin has an oxytocin has an isoleucine at position 3 and a leucine at position 8, whereas leucine at position 8 residue, and in vasopressin it is arginine. Here we have arginine, here we have leucine. Here we have position 3 isoleucine, here we have phenylalanine. Now, each of them is called nonapeptide, and both of these peptides have considerable physiological importance as hormones, and if you are interested, you can see biochemical connection.